Good morning everyone, have a great day. Today I am going to start a very important topic, that is the DNA. In my part 2 lecture of nucleic acid. Be attentive in the class and concentrate, my dear children. In this topic DNA, I will be discussing some of the important terms related to DNA, the structure of the polynucleotide chain. Along with that, I will be focusing or I will be discussing some of the important questions, probable questions of NEET and board examination. Okay, have a look towards the board, my dear children. Nucleic acid, that is mainly the DNA. The DNA is an acidic substance present in the nucleus, first identified and named as a nuclein by Fried-Rich mixture in 1869. Fried-Rich mixture. You can remember mixture in 1869. Very important question. This is the line of NCERT, my dear children. Clear or not? So all lines of NCERT must be by hearted, must be learned. Next one is DNA is a polymer. DNA is a polymer or chain of deoxyribonucleotides. Okay. DNA is the storehouse of genetic information. DNA is the storehouse of genetic informations in the form of the sequence of nucleotides or sequence of bases. Okay, children? DNA is the chemical basis of heredity. Question comes that chemical basis of heredity is, then the answer would be DNA. But physical basis of heredity, yes, can you tell me, my dear children in the class? Physical basis of heredity, yes, that is your gene. Gene is also called the unit of heredity. Remember this thing. But the vehicle of heredity, anyone from the class, my dear children, vehicle of heredity is chromosome. Clear? So some of the important questions may be asked. Very simple but important. Because NEET is now changing the pattern of the questions. Clear? The basic concept are mainly emphasized. The length of the DNA is usually defined as the number of nucleotides or nucleotide pair or base pair. Have a look towards the board, my dear children. A table is given, left hand side the name of the organism and right hand side the length of the DNA or the DNA content is given. Very important table is here. Now 5x174 bacteriophage having your SS DNA means single stranded DNA and circular DNA. As because it is single stranded, so the number of nucleotides will be there or bases will be there, not base pair. See the number. It is 5386 nucleotide. 5386 nucleotides in 5x174 vector. Clear or not? In lambda phase, that is 48502 base pair is there. In E. coli, the DNA content is 4.6 into 10 to the power 6 base pair. Of human DNA is your, that is your 3.3 10 to the power 9 base pair, my dear children. In this table, generally repeatedly two examples are being asked. The DNA content of 5x174 bacteriophage, very important. That is your 5386 nucleotides. Clear? Not nucleotide pair or base pair. Next, your lambda phase. Lambda phase is also called temperate phase. You have studied in standard 11. That's why the question is just twisted and asked, what is the DNA content of temperate phase? That is your lambda phase. Clear? Now children, some of the important questions are given on the board. You see, and just see the bottom of the board. Some of the important questions are discussed about the DNA. You see, DNA having both autocatalytic as well as heterocatalytic property. Is the statement is true? Yes. Then what is autocatalytic? DNA can form DNA. That is autocatalytic. DNA to DNA. That is replication. It is autocatalytic property. Now DNA to RNA, that is heterocatalytic property. Clear? So DNA having both autocatalytic as well as heterocatalytic property or function. Next you see the nucleotides of DNA, that is DAMP, DGMP, DCMP, and DTMP. D stands for deoxyribose sugar. That is adenosine monophosphate. That you can call it adenylic acid. I have told you earlier also. GMP, guanosine monophosphate, guanylic acid. I have told you. And similarly, cytidinic acid and thymidinic acid. Okay. Now these are nucleotides of DNA are active nucleotides or inactive nucleotides. These are all inactive nucleotides, my dear children. The DNA nucleotides are inactive. The statement is correct. Inactive nucleotides of DNA become active. Then the answer will be during the time of replication. So during the time of DNA duplication, replication, the inactive nucleotides of DNA get activated. Clear or not? Next is your, all these questions I have told you, and next we are going to the structure of the polynucleotide chain of the DNA.